Today we're going to continue with our topic of reproduction, and we're going to talk about sexual reproduction. So sexual reproduction is the process of creating more individuals of the same kind or same species. When we carry out sexual reproduction, unlike asexual reproduction, there are two parents that contribute genetic information. So the offspring are similar to the parents and each other, but not identical like they are in asexual reproduction. The process used for sexual reproduction is called meiosis. Sounds very similar to mitosis, but instead it's meiosis. If you think of humans being sexually reproducing organisms, I like to think of the I in meiosis instead of mitosis. I carry out meiosis. Meiosis creates certain types of cells that we call sex cells. The male will create sperm, the female will create egg. Another word for sex cells would be gametes, just a fancy scientific term for any sex cell created. So during meiosis, we have some differences compared to meiosis. During meiosis, the offspring or end result cells are going to have half the number of chromosomes as the parent. So instead of 2n, like we had during mitosis, they will only have n, or the haploid number it's called. Basically, it means they only have one copy of each type of chromosome. N stands for the different numbers or types of chromosomes you have. So in this case, we only have one copy of each type of chromosome. The other importance with meiosis is that we now have genetic variation because we are having two different organisms contributing DNA or genetic information to the process. Remember, in humans, we have a total of 46 chromosomes if we have the 2n number. We have 23 different types of chromosomes. That's our n number. And in the case of a haploid number, we only have one of each type of chromosome, which means we're going to have 23 chromosomes in our sex cells. As far as the chromosomes go, during meiosis, we inherit half of the chromosomes from each parent. So each parent's going to contribute half. So in humans, we would get 23 from the mother and 23 from the father to give us that 46, which would be 2 copies of each type of chromosome. Remember, during the cell cycle, we're going to have chromosomes that are going to be in pairs in our beginning cells. So we're going to use some abbreviations here to help you out. M is going to stand for maternal, mom. P is paternal, dad. I know the P and the D don't fit, but the M's all match. If you look at the drawing here, we have 2 from mom, 1 of this type, 1 of another type. So we 1 and 2 from mom, 2 from dad. Notice that there's also 2P and 2M, meaning that dad, paternal, gave me 1 copy of that type of chromosome, and the mother donated a different copy of that same type of chromosome. Same thing with chromosome number 1. Each chromosome contains a certain order of genes. So all of the chromosomes, number 1, let's say, would have hair for one gene, maybe eye color for another gene, and let's say skin color for another gene. All of the chromosome number 1s would have those genes there, but in terms of what you inherit from your parents, it might have a slightly different code. My hair color might code for brown hair as opposed to blonde hair. In terms of the chromosomes, they're paired. Remember, we have 1 from the mother, 1 from the father, so maternal and paternal, that have the same gene order, or the traits are in the same order on the genes. We call these homologous chromosomes. So all of the chromosome number 1s, or that type of chromosome, code for the same traits. The gene doesn't need to be made up of the same exact DNA sequence, or the bases, the A's, the T's, the G's, and the C's, may not be in the same order, but the actual genes, hair color, eye color, skin color, would be in the same order. I'd like you to pause and try for a minute. If you think about the fact that chromosome number 12M came from the mother, chromosome number 12, which chromosome would that be homologous to? And then chromosome 21P, what is that homologous to? Now that you've paused and attempted, we know that chromosome 12M refers to my mother. It's homologous to the same type of chromosome from the father, so that would be 12P. And in terms of chromosome 21P, that's paternal, so it's going to be homologous to 21M from the mother. We'll be practicing this as we go. Moving on, the actual process of mitosis is pretty similar to the process used for asexual reproduction, but in this case, instead of having one series of cell divisions where that one cell splits into two, we're actually going to have two series of cell divisions, and you'll notice that the process is very similar to that of mitosis. So these two cell divisions we refer to as meiosis 1 and meiosis 2. If you look in the beginning, it starts with an interphase, just like mitosis does. Notice we have the two maternal chromosomes, two paternal chromosomes, and during interphase we are replicating the DNA and we're preparing for meiosis. Notice that when we get to the end of interphase here and the chromosomes have made copies of themselves, we now call these sister chromatids. So we have the original and its copy, and this happens to be chromosome number 1 from the paternal side. Remember, they're identical to each other, the original and the copy. We move on to prophase 1. Prophase, the nuclear membrane is starting to disappear, and in this case we have what we call homologous chromosomes lining up. So all of chromosome number 1 are lining up. So the maternal and its copy and the paternal and its copy of chromosome number 1 line up together. So there's actually 4 chromosomes next to each other. We call that a tetrad. So the tetrads are going to form. These homologous or similar chromosomes are going to come together. During this phase, something pretty unique to meiosis can happen, and that's called crossing over. And what can happen is a small section of the chromosome from the maternal side next to the paternal side can accidentally kind of swap with each other. They're kind of sticky to each other, and they can exchange information, creating new combinations of traits that weren't seen before in that lineage. So we consider this to give us some genetic variation. That's one of the ways that meiosis gives us a lot of genetic variation compared to mitosis. So after prophase, where the chromosomes are pairing up, nuclear membranes disappearing, and in animals our centrioles are formed in the spindle, we end up with metaphase 1, where they're lining up in the middle, metaphase, middle. So they line up in the middle as tetrads. During anaphase 1, they're splitting apart, so the tetrad splits apart, so we have pairs going to opposite sides or poles. And then telophase 1 and cytokinesis, where the membrane pinches in animal cells. We divide up the chromosomes and also the organelles. So this was meiosis 1. This is our first division. Notice that in the end we're going to end up with, and we'll see it on this one, 2 cells. Now the chromosomes still have the original and their copy attached to them. So it still looks like we have 4 chromosomes here. We're going to see, though, that as we move on, we're going to end up with another division. So before we start meiosis 2, we're not going to have another interphase with DNA replication. So we have a second division without replication happening. So during meiosis 2, we go straight into interphase from that cytokinesis phase, or straight into prophase, from that cytokinesis phase. So now we call it prophase 2. These chromosomes are going to line up with each other, pair up with each other, the nuclear membrane is going to disappear, and now we don't have tetrads form like we did during metaphase 1. We just have the original and its copy lining up in the middle, and it is a relatively random order. Right here we might have 1 and 2 shown, but if we had multiple chromosomes, we might have chromosome number 1 here, or number 4, 7, and so on. They could be done randomly. They attach to the spindle fibers, and then during anaphase 2, the chromosomes split apart at the centromere, so now we have a single chromosome, not a doubled chromosome, and we're going to have each of those cells split into 2. Kind of looks like mitosis, kind of looks like that basic cell division, but now if you notice, each of the cells only has 2 chromosomes, and if you notice back to our coloring scheme, we have only 1 of each type of chromosome. That was one of the reasons why we lined up so nicely in the beginning before. We now only have 1 of each type of chromosome, and we've made sex cells, sperm and egg. This is nice because when the sperm and the egg come together, they will join their DNA. The sperm will donate 1 chromosome number 1, and the egg would donate another chromosome number 1 to get that pair again. So the end result, we have 4 daughter cells. They have the haploid number, or N number, meaning they only have 1 of each type of chromosome. So in the case of humans, it would be 23. We have sources of genetic variation being from crossing over during prophase 1, where the information can be exchanged with the tetrads. We have DNA that separates or segregates and recombines randomly. So we don't know which cell is going to get which set of chromosomes. We just know that they get each of the different types of chromosomes. So how they line up and pull apart ends up being random. So you can see that meiosis gives us a lot of changes. It's a relatively long process, but the nice thing about it is it's very repetitive. Meiosis 1 does mirror meiosis 2 very nicely. But at the end result, remember, we have one cell dividing into 4 cells with half the number of chromosomes as the parent cell.